Okay, before we start talking about meiosis, which is today's topic, here are 6 questions from previous lessons. I'm going to get you to just pause here, write down those questions and write down your 6 answers. And when you're done, press play and we'll talk through those answers. Right, so question 1 was, what is the function of ribosomes? And the answer to question 1 is that ribosomes manufacture proteins. So tick for that, or correct in your book. Question 2, what are gametes? And gametes are sex cells. So in humans and animals, they are sperm and egg cells. Question 3 asked you, what is the difference between a mixture and a compound? Well, in a compound, remember, the different elements are chemically joined together, and in a mixture, there is no chemical reaction. There's no chemical joining of the different parts of the mixture. Question 4, is salty water a mixture or a compound? Well, the answer to that is a mixture. Salty water is a mixture of 2 compounds. First of all, it's a mixture of water, which is a compound of hydrogen and oxygen, and salt is also a compound, which is sodium chloride. But salty water is a mixture of those 2 compounds because the water and the salt are not themselves chemically joined together. And question 5 was, what is the difference between speed and velocity? So speed is a scalar quantity because it has a magnitude, it has a size. So you might be traveling at 10 meters per second, that would be your speed. Velocity is the same as speed in every way apart from one, and that is that velocity must have a direction. So velocity is a vector quantity. So if you're traveling at 10 meters per second, that is your speed. If you were traveling at 10 meters per second west, that would be your velocity. And question 6 said, how is a constant speed shown on a distance time graph? Well, a constant speed on a distance time graph would be shown as a straight sloping line. It would have to be sloping because your distance, which is on the y-axis, would always have to be changing if you were traveling at a speed. So the only way you can have a flat horizontal line on a distance time graph is if you weren't moving at all and your speed was 0. So just take a minute to pause here and write down any corrections that you need to do to those questions, and when you're done, press play. So let's just remind ourselves of what we've looked at so far. We previously looked at mitosis, and you've got on the screen there the stages of mitosis. So the first thing that happens before mitosis actually begins is interphase. And during interphase, each chromosome creates a copy of itself. And as we move through the stages of mitosis, we end up with 2 daughter cells, and each of those daughter cells is genetically identical to our original cell. So mitosis can be used by the body to constantly create new copies of existing cells. I'd like you to take a moment here just to remember the 2 main purposes of mitosis. What 2 functions does the body use mitosis for? I'm going to give you 5 seconds to remember those 2 things, and then I'm going to put the answer on the screen. So the body uses mitosis for growth and repair. So we create new cells in order to grow, and we also need new cells to replace dead or damaged cells. And there are 2 really important bits of information we need to take away from mitosis before we move any further. The first is that human beings have 46 chromosomes in 23 pairs. 23 came from one parent and 23 came from the other parent. So we have 46 chromosomes. The other thing we must remember is that cells created by mitosis are identical to each other. The DNA in the daughter cells is the same as the DNA in the original cell. So let's just remind ourselves that in order to create offspring, living organisms have to fertilise or fuse together two gametes, one from each parent. So for animals, including humans, that's a sperm cell and an egg cell. And remember that we also said that each cell in the human body has 46 chromosomes. Well, if you take a sperm cell and an egg cell and they both had 46 chromosomes, then the cell created by fertilization would have 92 chromosomes. And each time that cell copies itself by mitosis, creating identical daughter cells, each of those cells would also have 92 chromosomes.
Hey, we've got a problem here.
And that creates a bit of a problem, because we know that our offspring will also have the same number of chromosomes as we do, which is 46. So fertilizing together normal human body cells, each with 46 chromosomes, won't work. The solution to this problem is a different kind of cell division, which we call meiosis. So before we talk through how meiosis works, let's recap. Sexual reproduction always involves joining together two sex cells or gametes during a process of fertilization. In animals, the male gamete is the sperm cell, the female gamete is the egg cell. When fertilization occurs, the egg and sperm fuse to form what we call a zygote. And we need the zygote to have the same number of chromosomes as each parent had. And for humans, that's 46 chromosomes. So each gamete must only have 23 before fertilization occurs. Almost every cell in the human body has 46 chromosomes, and these are called diploid cells, which comes from the word double, because they have 46 chromosomes in 23 pairs, one from each pair from each parent. Gametes, sperm cells and egg cells, have half this number and are called haploid cells. Once the two haploid cells, the gametes, the sperm cell and the egg cell, are fertilized together, the zygote then undergoes mitosis to form the embryo and eventually grow into the baby. So, gametes are haploid cells and are formed in the body by a process called meiosis. We can break the process of meiosis down into 4 stages. In the first stage, we start off with a normal body cell, just like we would in mitosis. And that cell is a diploid cell with 46 chromosomes in 23 pairs. Stage 2 is that each of the chromosomes replicates or copies itself, forming an X shape. Stage 3 is that that cell divides into 2 cells, taking half the chromosomes into one cell and half into the other cell. And in the last stage, each of those cells divides again. There are now 4 daughter cells, each of them a haploid cell with half the number of chromosomes that we started with in our original diploid cell. It's also important to notice that the daughter cells formed in meiosis not only are haploid, but they are genetically different from the original cell that we started with, unlike in mitosis, where the daughter cells are always identical to the original cell. Now, when fertilization occurs, our 2 haploid gamete cells can fuse together to create a new cell with the correct number of chromosomes. Your first task today then is to recreate the diagram of meiosis in your book, and then underneath that diagram or next to each section, make a note of the 4 stages that we've just talked about. So you can pause and rewind the video here and get that information and make sure it's in your book. Okay, on the screen now we've got an exam question. This is just a 3-mark question. So it's asking you to name the process in which the following cells are made: a zygote, an embryo cell, and a sperm cell. Okay, so pause here, write down those questions and write down your answer, and then press play when you're ready. Okay, so the first part of that question was name the process by which a zygote is made. And a zygote, remember, is the original cell that forms when a sperm cell and an egg cell, 2 gametes, fuse together. And that process is called fertilization. Part B was an embryo cell. An embryo cell is a cell that forms after fertilization has taken place. So we know that once fertilization has taken place, the embryo will grow by the process of mitosis. So embryo cells are formed by mitosis. And the last one, C, was a sperm cell. And a sperm cell, remember, is a gamete. And we know that gametes are haploid cells. And haploid cells must be made by the process of meiosis. So if you need to correct any of those, just pause here and do that.
There's a certain set of cells in the body that are quite different from all of your other cells. These cells are called gametes, and they contain half the genetic material that every other cell contains. And these are formed through the process of meiosis. If you look around at people, you'll see that, apart from identical twins, we all look quite different. And the reason for this is that we all come from different sperms and eggs. Only identical twins or triplets come from the same single fertilized egg.
Meiosis takes place in the testes of a male to form sperm. It also occurs in the ovaries of a female to form ova. These ova and sperms are collectively called sex cells or gametes. Meiosis gives rise to 4 different cells and is only involved in forming gametes, so that varied offspring result. Mitosis, on the other hand, produces 2 identical cells and is primarily used for growth. Each cell in your body contains 23 pairs of chromosomes, that is, 23 single chromosomes from your mum and 23 single chromosomes from your dad. The matching chromosomes from your parents have matching genes. So you have 2 copies of each gene. For example, let's imagine a gene for nose length. You have a copy of the nose length gene from your mum and another copy of the same gene from your dad. These copies are called alleles and may not be the same. The allele on the chromosome from mum may say long nose, and the allele from the chromosome from dad may say short nose. So you may have either a long or short nose depending on which allele is dominant.
A cell which is ready for meiosis has to have doubled up all of its chromosomes. By doubling up or replicating the chromosomes, the cell ensures that there are enough chromosomes to share around at the end of meiosis. If there are too many or too few, the resulting cells won't work properly, if at all. As the cell enters meiosis, it's producing an identical copy of every chromosome, so that it ends up with 46 pairs of chromosomes. The cell then divides, much like it did in mitosis, to give rise to 2 cells with 46 chromosomes each. The cell now divides again to form 4 varied daughter cells with only 23 single chromosomes in each. The cells are varied because it's entirely random which chromosome goes into which cell. To go back to our nose example, a cell may have the chromosome with the allele for a long nose, or it might have the chromosome with the allele for a short nose, but it won't get both.
You now have your 4 gametes with 23 single chromosomes each. These cells are referred to as haploid cells because they have half the number of chromosomes. The original parent cell was diploid, which means it had 23 pairs of chromosomes. The haploid gamete cell has to wait until it fuses with a haploid gamete from the opposite sex. Once this has happened, the new cell formed from the 2 gametes has 23 pairs of chromosomes. In other words, the normal diploid number of chromosomes is restored. This time, the chromosome containing the allele for short nose may find its matching chromosome has the allele for a medium-sized nose.
The new cell formed from the fused gametes is called a diploid zygote. It can eventually grow into a baby through the process of mitosis. As you can see, if the original gametes contain the normal number of chromosomes, then when the gametes fuse, they would make a cell which had 46 pairs of chromosomes. Such a cell simply isn't viable, and this is why gametes are formed by meiosis, which reduces the number of chromosomes by half. In short, meiosis results in the production of 4 haploid, non-identical gametes. These gametes are called ova in women and sperm in a man. Meiosis promotes genetic variation among individuals of the same species.
Okay, well done for your work today. Your final task for this lesson is to complete the quiz, and you can access the quiz by clicking on the link underneath this video in the description. Remember, that quiz will get sent to me automatically, and you must complete the quiz in order to have completed the lesson.